Hello, everyone. Welcome to Brainmap. This seminar series is co-sponsored by P41 funded Center for Mesoscale Mapping, housed in the Martinos Center. Before we start, I just want to celebrate everybody's International Women's Day and remind that there is a stand on the first floor for women in science. Please just go and visit them and show your support. And today, as the day I think supposes, we have a female speaker, very accomplished one, and welcome Dr. Poornima Kumar. Dr. Kumar completed her bachelor's in instrumentation engineering at the University of Madras, India. She then completed her master's in medical imaging and PhD in mental health at the University of Aberdeen, Scotland. Following the Dr. Kumar finished a postdoctoral fellowship at University of Oxford, she joined Center for Depression, Anxiety, and Stress Research in 2011, where she completed an additional postdoctoral fellowship with Dr. Diego Pizzagalli. Poornima Kumar is currently an assistant professor at psychiatry in the Department of Psychiatry at Harvard Medical School, and director of computational and psychopathology group at the McLean Hospital. Her research program focuses on understanding how we make reinforcement learning based on decision making and how this goes awry. That's a very, very interesting word. Awry in individual and psychopathology specifically, mood disorders. Dr. Kumar uses multimodal assessment, MRI, EEG, behavioral tasks, and pharmacological manipulation and computational models to elucidate the neuronal, behavioral, and molecular mechanisms of reinforcement learning. So I would like to remind our audience, please address your questions for Zoom by raising virtual hands or Q&A box at the end of the talk. And for our individual in-person people, just raising your hands at the end of the talk. Dr. Kumar, thank you very much. I'm very excited to hear your talk. Thank you.
Thank you. Okay. I'm very happy, very excited to be here, especially on International Women's Day. I didn't know until I was on Zoom about this morning. And thanks very much for the invite. It's a long journey for me to get from McLean Hospital here this morning. It's nearly 10 miles. So brutal journey, just kidding. But I'm very excited to share my research and talk about new collaborations and new projects, etc. Okay, so I'm going to today talk about reinforcement learning and how the brain regions involved in this behavior and how we can use RL to understand heterogeneity of psychiatric disorder. So what is RL? Reinforcement learning is the critical process of survival that is observed across many species, including humans, where we learn by interacting with the environment and you learn from one's consequences and adapt your behavior to maximize rewards and minimize punishments. Learning is said to occur when there's a discrepancy between what you expect and what you actually got. When the outcome is better than what you expected, you get a positive prediction error. When it's worse than you expected, you get a negative prediction error, and it's same as what you expected. There's no learning set to occur, and there's no prediction error signal. There was this instrumental paper in 1997 or so by Scholes, where they showed that the dopamine neurons of the ventral tegmental area, substantia nigra, and the midbrain encodes this prediction error. So this is I should use my mouse, I suppose. This is a recording of the neurons, the dopamine neurons in the midbrain, ventral tegmental area, and in a monkey. And what happens here is at the top shows the average response of the dopamine neurons. So when an unexpected reward is delivered, so the animal doesn't know that there's going to be a reward, and you see this kind of firing of the dopamine neurons at the time of the reward, corresponding to a positive prediction error. And during several trials of learning, for this example, the Pavlov dog was learned to associate the bell with the food. Like every time the bell was rung, the food was delivered. So the animal learned that there was an association between these two stimulus. And after several trials of learning, at the time of the reward, the animal knows that it's going to expect a reward, so there's no prediction error, so there's no firing at the time of the reward. Now imagine you withheld the expected reward, so there's going to be this negative prediction error, so you see a dip in the firing of the dopamine neurons corresponding to a negative prediction error, and of course the dog is very sad. And what's interesting here is at the time after learning, even though there's no prediction error at the time of the reward, the dopamine neurons fire backward in time, meaning that they fire at the time of the conditioned stimulus. In this case, the dog drools at the time of the bell rather than the food itself. So you start to kind of move backward. This is similar to maybe we have association from childhood memory, like ice cream ban, we hear the bell, you kind of think about, you have to elicit the pleasure response. You don't necessarily need to have the response or to have the reward. So just to kind of briefly, reinforcement learning is you have a prediction, you interact with the environment, you pick the action, you get the outcome, based on the outcome, you update your prediction of the stimulus that you choose. So how do we kind of quantify this complex behavior and make sense of this? That's kind of where computational modeling comes along. And just to give you a brief idea about what computational models are, you take any kind of behavior, and we use mathematical models to explain this behavior. And we usually have like several different models to understand the behavior and figure out which mathematical model explains the behavior best. And once you figure that out, you then extract subject-specific behavioral parameters, which you can then use on to identify individual difference by mapping onto different phenotypes of brain circuitry, etc. So computational models are very powerful compared to traditional statistical methods because it helps us identify hidden constructs of behavior. So if just looking at what choice they picked, you get an idea, but you're missing out like how they got to pick that choice, how they learn. The dynamics of the learning is missed if you don't kind of use these kind of models. It helps us interpret complex data and generate new hypotheses, etc. And the field of computational psychiatry has kind of just exploded in the last decade or so, or last even few years. This is just a very simple review paper search on PubMed using the keywords computational psychiatry. So when I did my PhD in around like 2007, 2008, there was hardly any paper on computational model, like one or two. And if you come to the clinical population, there was hardly any. But you can see just the amount of review papers on this field is kind of just exploded in the last couple of last few years. And the field is kind of adapting or understanding the strength of using computational approaches to understand this complex psychopathology rather than using more conventional approaches. And if anyone's interested, there's a really nice paper by Wilson and Collins. It's called something like 10 Simple Rules to Build a Computational Model, where they go through how you would design a model for any experiment, how we validate it, and come up with the hypotheses, etc. Okay, so based on the computational model, you get some behavioral parameters, which I'll explain to you in a second, which are more like explained by how they perform during a task or how they use the feedback in the environment, etc. And you also get some parameters which tells you about how they got there, the dynamics of decision making, for example. So if you go back to a simple flowchart again, for example, you have a prediction, and as I was saying, you get the outcome, and then you update the prediction based on the prediction error. So the rate at which we update this value, it reflects on this parameter called learning rate. For example, it varies between 0 and 1. So for example, if your learning rate is 0, so this term goes away, so the value of your prior or value of the current stimulus is exactly as the prior stimulus. So meaning that you don't care what happened just now, you care about what happened in the past. And on the other hand, the flip side, if the learning rate is 1, you give a lot of importance to the prediction error, meaning that even though it gave you what happened in the past, just immediate prior to the current trial, whatever happened. So you can manipulate, or individuals vary how much feedback information you want to include in updating your behavior. And the second parameter, for example, is called the noise parameter, beta, which reflects to the randomness in your output, how much you exploit or explore, for example. Again, this balance is very critical. in human beings. Sometimes exploiting is good because it's safer, you know what's out there, but sometimes exploring is good because if you don't do it, you don't know what's out there, right? So the balance between exploration and exploitation is also critical and is impaired across different psychopathology. For example, individuals with impulsivity or in manic episodes tend to seek more rewards. They tend to explore, even though the reward might not be that rewarding or the value is much lower, they will still kind of tend to explore more choices, which might be detrimental, more costly. On the other hand, individuals with depression tend to exploit more, stick with the past experience. They don't want to try new things, or people with anxiety. And the other parameter is called salience attribution, like how much you like a particular reward. So we all know everyone would say, oh, I love going to that restaurant, but how you rate it might be very different. So that kind of, this parameter again varies, like how much you enjoy the particular reward. And of course, the other two parameters of prediction and prediction error is the dynamics of the learning rate. So you can get all of these different parameters, which you wouldn't get if you just look at average responses to reward versus punishment, etc. So that's the kind of the power of computational models, is like trying to parse these individual processes. So using this kind of approach, again, consistent with the dopamine study I showed you, human studies have been kind of shown to have prediction error signal in the ventral tegment area and the striatum and the prefrontal cortex, which are rich in dopamine. And many studies have kind of, again, with respect to reward, it's been kind of shown, but on the flip side about how we learn about punishments has been a little bit less studied until a kind of a decade or so ago, where they found maybe the GABA neurons of the habenula, which is like a really tiny region, might be encoding the punishment prediction error, same as the BTA. But what they found was the BTA and the habenula neurons are kind of acting like a seesaw, for example. So just showing this kind of graph, the blue line corresponds to when there's a no reward or a punishment. The habenula neurons fire, and the idea is it then inhibits the dopamine neurons in the BTA, which reduces the dopamine release into the striatum prefrontal cortex. On the flip side, at the time of the reward, the habenula neurons pause firing, which disinhibits the BTA dopamine neurons, and thereby increasing the dopamine releases. The idea is it acts like a seesaw. One goes up, the other one goes down. And consistent with this, we've shown in a meta-analysis, and other people have shown, like reward prediction error in the striatum, punishment prediction error in the habenula, etc. So what we want to do at this point, this goes back years during a PhD, is like, can we understand depression using reinforcement learning? For example, core features of a clinical depression is, one of them is anhedonia, the inability to experience pleasure or reduced motivation to seek pleasurable activities. And on the other hand, you have these negative bias, which is more associated depressed mood, guilt, rumination, etc. So maybe one of the questions we wanted to ask is like, can this features to be explained by abnormal reinforcement learning? Could anhedonia be associated with reduced reward learning, and negative bias could be explained by increased punishment learning? Okay, so one of the first studies we did, we looked at healthy controls and individuals with depression, and what we found was P represents the patients had a decreased reward prediction error signal in the striatum, consistent to our hypotheses. But what was one of the limitations for this study was all the individuals with depression were on long-term medication. They were taking antidepressant for years, so they're almost like treatment resistant. So one of the questions was like, is this reduction really due to the illness or due to the medication? The reason we were thinking that was when we gave antidepressant to healthy controls for 3 days and we scanned them, we found a reduction as well. So this is always like, is a treatment, as a confound, medication confound. So several years later, I was able to do this, repeat similar study in a unmedicated depressed individual. And in this one, so this study, we used drops of water as reward. So we asked them not to have any water since dinner, and we scanned them first thing in the morning. So they'll be in the scanner with the tube, and so they'll be thirsty. So any drops of water is rewarding. And in this case, we used money, where again, 2 pictures, one of them associated with more money than other, and they have to decide which one to pick. So here, what we found, again, we looked at the striatum because this was the regions that's shown in the meta-analyses. And what we found was, again, consistent with our original study, we found the prediction error in the striatum was decreased in individuals with depression compared to healthy controls. And we also wanted to now look at the ventral tegmental area, because that's kind of where the dopamine neurons are encoding the signal, and we didn't find any difference in the prediction error strength. But what we found later was the connectivity between these two regions were abnormal. Meaning the MDD individuals, depressed individuals had a reduced connectivity between these two regions, thereby which kind of made us think maybe the dopamine neurons are encoding it appropriately, but it's not being communicated to the regions, downstream regions for decision-making process. And this was further reinforced by our prediction error signal in the striatum, also correlated with the connectivity strength, but also correlated with the prediction error of the ETA, which was kind of really good. And on the other side, we were also able to look at the punishment kind of thing, and where we found no group difference between healthy controls and depression, but we said overall punishment prediction error signal was higher in Athinoula, which was kind of good. But what was really interesting was both the reward prediction error and the punishment prediction error correlated with a number of episodes, meaning that higher the disease burden, there was more of a deficit in the learning in our population. Okay, so further along, we were able to show that this prediction error signal in the same region, the striatum, correlated with self-reported depressive symptoms, but also correlated with prediction error calculated in the real world, like using smartphone. We asked them questions like, what are you looking forward to it? Did you enjoy, etc. So that was kind of really nice. We were able to replicate it in another study, another population, another task. And it also was correlated with genetic risk score, meaning if you have a higher vulnerability to depression, they had a more or an impaired or less adaptable to when dealt with acute stress in the study. So the prediction error signal was associated with a higher risk score of depression. So all this is kind of great. So this has kind of understand like, okay, depression could be associated with reduced reward prediction error signal, and there's a potential punishment angle, but more work needs to be done and the sample size is small. And the other, also thinking about reinforcement learning or any decision making is not simple as picking two choices and you're getting a reward. It's much more complicated. Even a simple decision like picking a burger or a pizza for lunch involves lots of decision making process. You might have to think about how far it is the burger place or how good the pizza was last time when you had with your friends. And so we constantly do these cost benefit analyses. Even though we are not aware of it, we are constantly making these computations. And you pick your choice based on the choice you explore or exploit, you get the outcome, you update the behavior. So it's lots of computations happening even in a simple decision-making task. And luckily, we have computational tools, algorithms to explain each of these sub-processes, which gives us more estimable parameters that help us kind of understand individual differences and stuff. These are just kind of some of the parameters that have been defined for the reinforcement learning task. And if you look at the brain side, which is what we are kind of interested about, is not just that, but also interested about is there's also never one-to-one mapping with a construct. There's no one mapping between one region and another construct. Is usually multitude of regions are involved in a particular construct, often overlapping with other constructs. And sometimes you have distinct patterns too. And what we know about the circuitry of reinforcement learning is not as pretty as this picture. This has been put together by lots of preclinical work and some human work and stuff, but very few studies have looked at all of these together to get an idea about the more network approach. So the very first question, before we can kind of go on to identifying biotypes or biological targets, etc., we need to fine tune this model and identify more better tune the neurobiological model so that we can kind of then develop treatments or interventions, etc. So there are two ways that we can kind of, or at least I'm approaching is like use structural connectivity, but also the functional connectivity, and use it to guide each other to help us tune the biological model. So the first, on the left, the structural connectivity, is actually, I'm working in collaboration with Chiara and Anastasia, who are very well known in this center, is to identify, validate existing and identify novel tracks that are involved in reinforcement learning decision making. So using kind of the tracular approach, which has been validated to kind of to manually label the tracks of interest and use them as a training set and use that to improve the automated reconstruction of the lower quality data. This is just a figure I took from one of Chiara's papers that shows some of the tracks are correlated with these MFQ and depression, like depression, anxiety ratings in an adolescent sample. So what we want to do here is extend this further to add other relevant pathways that are involved in reinforcement learning, like the stria terminalis or medullaris or fasciculus retroflexus, which are regions connecting brain regions that are more involved in reinforcement learning, like the BTA, Athinoula, medial prefrontal cortex, etc. So this figure is a beautiful figure is by Chiara put together for me for a grant, which shows these tracks in one subject, just as a proof of concept that we are able to see these tracks in our data. But of course, there are tracks like the one on the right, which are the dark blue connecting Athinoula and BTA, are very small that you need really high resolution data to even be able to see it. So the idea is once we can kind of identify these tracks, we can then look at the metrics, the white matter integrity metrics, and see how they are associated with different reinforcement learning constructs and how they could go awry in individuals with psychopathology. And in this collaboration, apart from working with lovely people, I get to say these fancy words like fasciculus retroflexus, which makes me sound very intelligent. Anyway, yeah, so that's the structural connectivity side. And on the other side, to identify the functional connectome, I use build dynamic causal models to help us identify effective connectivity between these regions. I use these new approach, the whole brain regression DCM, which has been developed in the last kind of few years, where you can identify a whole brain connectivity profile for an individual for a particular task. So the way this one works is like you start with a fully connected model. So it assumes every region is connected to every other region, and it prunes all the non-significant connection. So this way, I can identify networks that are involved during a task without any a priori model. And this approach has been applied to both task and rest, and there's some test retest reliability data available now on like a bigger data set. So using this method, I have a talented student who actually presented a poster on this in the Harvard Psychiatry Day, where we looked at what the connectivity profile during reward and punishment learning. I don't want to read too much into this because this is just a proof of concept on like 10 people or so. But the interesting thing is I want to draw your attention to the bottom two, where it shows the connectivity pattern or profile looks very different, reward to punishment versus punishment to reward. And this is really interesting. And we are trying to kind of dive into this and explore this and see how we can use the connectivity profiles to help us identify more regions that are involved during learning. Okay, so any questions so far? I know we said less, but no. Okay, so what we want to do next is, this is all good. So this is the first step to identify the connectome. And really the next kind of the theme of my research program is like, how do we use reinforcement learning and how do we kind of understand the heterogeneity? This is a big issue across all the clinical research, is like, how do we tackle heterogeneity? So even if you go back and think about major depressive disorder, so there are like several symptoms which are listed on the left, but for a clinical diagnosis, you need to have 5 or more symptoms for at least 2 weeks. So 2 individuals, for example, can have different symptoms, but they can have the same diagnosis. On the other hand, 2 people can have same symptom profile, but they have a completely different treatment response. And one of the studies which run in 2015, kind of showed that there could be 227 unique symptom profiles in this clinical MDD population. So this is very worrying, right? Like even the studies have shown in the past, usually sample size is like 20 or 25. So ideally, each one of them could have a completely different symptom profile altogether. And that's kind of worrying. So we need to kind of, there's obviously there's lots of issues that we need to address in psychiatry, but with heterogeneity, so at least we need to start thinking about collecting big data and better approaches to tackle this. So also, on the other hand, reinforcement learning has been also associated with different clinical phenotypes. And most of these studies are all, the evidence is mixed. Some studies find it, some studies don't find it. Again, it's because of heterogeneity, it's because of sample size, etc. So this is just another talented student. We have collected a lot of an online data set from about 2,000 individuals during COVID time. And they did a lot of mental health questionnaire and they did a social RL task. And this is just to kind of show you that the mental health questionnaires are mapping onto different parameters. They're all over the place. They're not the relationships are not that strong. These are all significant findings, by the way. But this is just to kind of show you that it's all over the place, what we know right now with mapping of reinforcement learning and phenotypes. So one of the approaches framework NIMH put together to identify these homogeneous biotypes, which we need to identify biological target information, is called RDoC. I don't know whether people know about this. This is a framework that they put together to encourage researchers to measure and integrate across different units of analyses, rather than just looking at one measure, like try to use different units of analyses. And we often measure a lot of data, like in any clinical research or any research, we just don't do one measure. We do multiple surveys, battery of tasks and different brain measures, etc. And the way we integrate, usually, it's we analyze them separately, map onto phenotypes, and try to make sense of it later on. But to truly integrate, we need to start thinking about linking these measures even before we make any assumptions about or mapping onto the phenotypes or diagnoses or etc. So there's been like several methods, or people are working on it to try to link different data types like self-report, GPS data, brain data, etc., to truly kind of get this multimodal pattern to help us parse the heterogeneity. But what we can do effectively now, and is being done across multiple studies, is to integrate brain measures. And lately I've been using this kind of multimodal imaging data fusion, which kind of the method that combines data from different modalities to identify these multimodal brain features. So the idea is it leverages this cross-modal information. If you look at them separately, you're missing out the kind of the significant cross-modal information you would get between these different modalities, which you would probably lose out where you're looking at them separately. And the important point about this method is it links the data at the point of the statistical analyses rather than post-hoc, which so that it better models the multimodal covariance between and within subjects. So I've been kind of trying to use this to look at some of the multimodal brain data we have and how we can map it onto reinforcement learning, or how we can use this method to identify more brain networks associated with RL. So I'm going to show you two studies just as proof of concept to see how this one works. So the first study is just a simple, again, a two-arm banded task where you see one or the other. One picture is associated with reward more often than the other. They have to decide which one gives you the reward at that particular time. And I use hierarchical Bayesian models to estimate different parameters corresponding to reinforcement learning. And this is so these are the two constructs I'm focusing on in this study. And it's a small sample size, it's only like 46 individuals. So for this, because it's a small sample size, we used only the structural data. Again, it's the T1, where we got the cortical thickness, PL surface area, and the gray matter density for every subject. And you put them all into this data fusion algorithm. And what you get is these multimodal brain patterns. So in this case, we got 11 because it's a small sample size. So this multimodal brain pattern could be truly multimodal, meaning that it can have contribution from all the brain features you're inputting in, or it could be more weight to one or the other feature. So once you get these patterns, you get these subject loadings, which are just the strength of the pattern in every individual. And after that, you could just map it onto phenotypes or do clustering to identify biotypes, anything. The world is your oyster. So for this particular study, what we found was we had 3 different reinforcement learning parameters. We got unique brain feature or multimodal brain feature associated with it, which was very interesting. Again, it's a small sample size, but it was really nice to see the separation between the three parameters. And on the right, it shows just the contribution from every one of the brain feature or the modality we put in. And you can see the first 2 are weighed. The first 2 are weighed towards one or the other. In this case, it's a PSA, and this one is the BBM. But the last one, we could kind of say is truly multimodal because it's got contribution from all 3 features that we put in. Again, small sample size, we need to replicate it. And I had another study option to capitalize on these publicly available data sets like the Human Connectome project, where there are about like 1,000 or so individuals who do a lot of brain measures. And then they did this delay discounting task outside the scanner. So if people don't know what delay discounting task is really it captures the rate at which we value future reward compared to now. So they're shown 2 options. You get $10 now versus, I don't know, $20 if you wait for like a month. So it depends on, some people might prefer to wait for 1 month or for now. So again, the rate at which we value the reward or the decrease in the value of the future reward varies across individual and is shown to, again, impaired across different psychopathology. For example, people with impulsivity are correlated with higher discounting, that meaning that they want it now, they don't want to wait. But individuals with anxiety tend to wait for more future rewards. So in this one, because it's a larger sample size, I also and it's publicly available, I also have got the option to include the DTI measures. These are like white matter integrity measures. So again, as I was saying before, we throw them all into the data fusion algorithm. You get these multimodal patterns, you get the subject loading, and then we can see how it maps onto different phenotypes. And we found 5 multimodal pattern correlated with this delay discounting factor. I'm going to only show 2 just for purposes of the talk. And the first one is the DLPFC kind of pattern. And DLPFC is interesting because this is one of the regions that kind of shown again and again in the delay discounting computation. And it's interesting to see the true multimodal pattern at the bottom, to a contribution from different features. We input it into the data fusion. And the next one is a striatal, my favorite region. Again, you can see the nice multimodal contribution from different features. So using this kind of approach, we can identify like large brain networks that have both biological and behavioral validity. And once again, as I was saying before, once these subject loadings are obtained, you can then conduct clustering to identify homogeneous biotypes. And these are interesting because these are the loadings that have been shown to be associated with a reinforcement learning parameter. And then when you kind of then take it to, we can, after that, it's up to us what we want to do, like go for intervention. Maybe individuals who are more impaired across learning rate might be better suited for behavioral activation versus antidepressant, etc. So the key about this approach is the better interpretability, meaning that once in the traditional approach where we have individual analyses and then we have to link, gray matter is associated with, I don't know, learning rate, this white matter measure is associated with what sensitivity, and then you're trying to make sense of it all together. But with this kind of multimodal data fusion, it does the fusion before, so you don't have to worry about kind of making sense of it later, basically. Okay, so that's pretty much all I have, and I would like to thank kind of my granting, the agencies and my mentors. And Lisa Nickerson is the director of Applied Neuroimaging Statistics Lab in McLean, and she's the imaging mentor of all the data fusion, and of course all the students and the participants. And thank you for joining, and also via Zoom and in person. And if you have any questions, feel free to talk.
Thank you very much for this great talk, Dr. Kumar. I just want to remind everyone that please ask your questions by raising your hand, or for the, that's me, for the virtual audience by typing out their also questions to Q&A box. In the meantime, when people are preparing their questions, I will use this time to actually ask my question. At that point, actually, I had a question. I was just a bit too late. So you showed the functional connectivity maps, right? I mean, it might be a very stupid question, but what I was thinking was they are quite different. So, and I could see that they might be kind of correlated with different, I don't know, states of clinical states and used as biomarkers, possibly.
Yeah, I think that's we don't know. I think they look different, but they represent just like, it could be just the strength of connections are very different across different during different processes, right? So that idea, yeah, we will now kind of go into and explore it further to maybe associate like different either symptomology or disease burden, or it could be just like having coffee. Yeah. Because again, these connectivity patterns, or just motion. I mean, probably it's all garbage, right? So I think that's what we are trying to do now, to explore it, to make it more robust. Yeah. Before we go on interpreting and changing the world.
Yeah. It was exactly the same for the last slides. Actually.
Yeah.
So I was like, can you use this?
Yeah, I think that's the future goal. But yeah, I think that before we do all of that, it's how stable these patterns are and these great features are. So those are, we need like more data, more just reliable. So, yeah.
Yeah. I have another question actually. For reinforcement learning, what I always think is it is a cause. Sorry, not a cause, but a consequence of. Could it be also a cause for some of those clinical symptoms?
Yes, because if you don't know, again, it could be the behavior is due to the illness, or the illness is causing the behavior. You don't know, but since the similar neuron, the brain regions involved in behavior patterns, we don't know the cause and consequence. But that's why we need more studies to look at longitudinal samples and at different states, etc. Yeah.
Okay. Are there any questions here? We have a question then from our actually virtual audience. Dr. Walid Yassin says, that's a fantastic talk. Could you elaborate more on the data fusion method and how it compares to PCA?
Yeah, if I can. So the data fusion is like ICA. So where the PCA is the first step before you do the ICA on them. So in a way, it includes PCA, but that answers your question. I don't know, but yeah, data fusion is just, if you think about independent component analysis. So instead of doing it for one modality, we put them all together. We do ICA on the multimodal features. So you would do an intermediate step, is like you would do the PCA to throw away the noise components before you do the ICA.
Okay, thank you very much. If nobody has any more questions, I will thank you very much. That was very interesting talk. And for our audience, see you next week. Thank you.
I can start.